So my name is Nate Weisenberg. I am here with the Atomic Heritage Foundation. It is April 25th, 2018, here in Oak Ridge, Tennessee. And I have with me Lee Russell.
Right.
And my first question is if you could please say your name and spell it for me.
My full name is Liane B. Russell. It's Liane, and the B stands for my maiden name, which is Brauch, B-R-A-U-C-H. Russell, R-U-S-S-E-L-L.
Great.
Okay?
So my first real question is, can you tell me when and where you were born?
I was born in August 1923, and I was born in Vienna, Austria.
And what did your parents do?
My dad was a chemical engineer, and my mother was a housewife. And I'm the oldest of three kids. I had a sister 2 years younger than me, and a brother who was 10 years younger than me, and ended up having 9 kids.
And what was your childhood like growing up there?
What was my childhood like? Very good, until we had to leave. It was, I was about 14 and a half when we left. So, you know, I had my childhood and early teens in Austria, and then first went to England for several years, and then from England came to the States. This was during the war, so shipping was very greatly restricted because of U-boats in the Atlantic. So, and it's very hard to get any kind of passenger transportation. So we traveled in a Cunard, what used to be a liner, but during the war they used it to go down to Argentina and bring back beef to England, and frozen beef. And so on the way down to Argentina, they went empty and took passengers, a few anyhow. So that's how we went from England to Argentina, and then picked up an American liner and came up from Argentina up to New York. It was a pretty adventurous trip.
Yeah. And did you leave Austria because of the Anschluss?
Yes, we did, yes. And we're very lucky that we were able to leave very early. We left just 5 weeks afterwards. So we left in, I think it was March of 1938. No, it could have been April. Anyhow, somewhere early in the spring. And went via Prague and Brussels to London. And then when the war broke out, which was a year later, our school was evacuated to a place called Berkhamsted in, I've forgotten, it was very close to London, about an hour from London, north of London. And we were evacuated and lived there with a local family for the rest until we left in 41. Yeah. So we left before America got in the war, because when we came here, there was no war here. You know, it was very strange coming from all the bombings and everything. But in December of the same year that we arrived, of course, Pearl Harbor happened. So it was a short time between wars, sort of. I went to Hunter College in New York, and then graduated in January of 1945, and went to graduate school at the University of Chicago. But in the meantime, when was it? Yeah, in 1943, I started a summer program. I attended a summer program at the Jackson Laboratory in Bar Harbor, Maine, and just happened to luck into it. I saw an announcement on a bulletin board at Hunter, and just responded to that and got accepted, and I went up there and spent the summer of 43, and it was an apprentice research program. In those days, it was very rare to have summer programs for students. Nowadays, everybody has one, you know. But so that was in 43, the summer of 43, and then the summer of 44, I went back there, and then after I graduated in January of 45, I went back as a technician and worked there until I entered graduate school. So.
How did you know or how or when did you know you wanted to be a scientist?
Oh, just as soon as I was going to say as soon as I entered the summer program, but before that too, because I think my dad always hoped I would be, and I was sort of steered in that direction. But, you know, because he was a chemist, and I didn't know I was going to be one. In fact, for a while I was going to be a writer because in my first year at Hunter, I took a writing course, and we were encouraged to send entries for the Atlantic Monthly National Essay Contest. So I sent an entry, and it won first prize, and so immediately I got called by all sorts of magazines and everything. And for a while, that's what I was going to be. After that, I was going to be an MD, and I actually took courses, you know, both chemistry. I had a chemistry major and a biology minor, so I had enough for entering med school. But I got talked out of it when I went to the summer program in 43. I really got bitten by a research bug, and that's what happened.
I think I had read that you saw, was it a mouse embryo under a microscope?
Yes, that was one thing. We had what we call tutorials. Different staff members gave different sort of small courses, and in one of those, I was looking down a microscope and I saw a mouse fertilized egg, and I thought, my God, 3 weeks from now, less than 3 weeks from now, this is going to be a mouse, a complete mouse. And that, you know, that really was an amazing thought. And that sort of got me turned on.
In Maine is where you met your husband.
I met my husband there, yes. Yes, he was the organizer of the summer program. So, you know, he had a lot to do with the students and with the researchers that supervised the students. Yeah. And
so you worked there for a few summers.
From June of 43, off and on, you know, with the year out for college. And I went from there to the University of Chicago. So from January 45 till September 45, I was working at the lab as a technician, and I was living up there, you know. So.
And how did you decide to come to Oak Ridge?
I didn't decide, but my husband decided to change jobs. So he was looking for jobs and getting various interviews, and Oak Ridge was virtually the only place where I would be allowed to work also, because all the others were university jobs. They all had nepotism rulings, and I couldn't have worked. And so I was able at Oak Ridge not only to work, but to do independent work. And, you know, that's why we came to Oak Ridge. That's why he picked Oak Ridge. And we came in November of 47. So we've been here, how long does that make it? 70? Yeah. Almost, yeah, over 70, because it was 47, okay?
71 years.
Yeah.
Wow. And I read about it in the article about a gentleman named Alexander Hollander.
He had just started at the, it was then called the Clinton National Lab. And, and he had just started a biology division. Before that, they had some biology experiments, but no organized division. And so in 46, I think, Alex Hollander started the biology division, and he was very anxious. He came from University of Wisconsin, and he had worked with ultraviolet light, and he was, I guess you'd call it a biophysicist, and so he was very anxious to people the new division with mostly with geneticists in various areas, Drosophila, Neurospora, Cytoscantia, you know, anything, but primarily geneticists. And at the time, I guess it was the AEC at that time, predecessor of the DOE, was very concerned about fallout from nuclear tests and the effects of fallout on people's health. And so they were anxious to, well, they started various health programs, but particularly they didn't have anything on hereditary effects of radiation. And that's what Hollander was offered essentially to establish at Oak Ridge. And so Bill was hired for that. And I came along.
And there had been a forest fire by the Jackson Laboratory.
Exactly. We were supposed to come earlier in the fall of 47, but in October there was a major forest fire which burned up almost half the island. You know, it's a large island, and it burned almost half of the east half of the island. It didn't really spread to the west. You know, Somme Sound divided the island into almost into two parts, and it didn't spread to the west. But the lab was on the east side, and it really, really perished. I mean, at that time, it was just a single building, but it was, well, not only was the building destroyed, all the wood, it was brick, but everything wood in there was burned up, and all the mice were asphyxiated. So Bill had gotten some mice together to come down to Oak Ridge, and of course they died too. So when we came down, we had no mice. We delayed our trip a little while because we were continuing to help and fight the forest fire up there, which did not end with the burning of the lab, but you know, it went on after that. And stayed probably October, no, 2 or 3 more weeks, I think, before we came down. Yeah.
What were your first impressions of Oak Ridge when you got here?
Bad. We drove down here. We left Maine in a snowstorm. It was a snow blizzard. It was blowing this way, right at the windshield, you know, couldn't see a thing. Anyhow, we left in a windshield, snowstorm, blizzard, and of course we got, the weather was good. after that. And we entered Oak Ridge through what is now the Clinton Highway, came in from the east end, and the whole stretch between what's called Elsa Gate, the east gate to Oak Ridge, to the so-called guest house, which is the only hotel facility. That whole stretch was built up in what we call hutments that were used to house the people who were building the labs and the town. And they were miserable looking things. They were like plywood boxes. They had no windows, they had shutters that you had to open to get air. They had no plumbing, they were separate bathhouses. They had one central little pot-bellied stove, they were really bad. And that's all I saw coming from the east entrance all the way to the hotel. We were going past these miserable-looking hutments. And I didn't have a very good impression of the town. But, didn't take more than a day before we started looking around. But it was different, but not that different.
And so, because of the fire, how did you get new mice for the laboratory?
Yes, that was a big problem because we needed not just mice, but very special mice. And we were competing with the Jackson lab, which was also trying to build up mouse dogs. And of course, they were well known, and we were not known at all because we were just starting out. So it was quite a job. And then we discovered, I don't know how, but there was a pharmacist in Central or West Florida whose name was Mr. Holman. And he had a hobby. He was a mouse fancier. And you probably don't know what that is. It was actually a hobby mostly during the 19th century. People would raise pretty mice, to make sort of a zoo of beautiful looking mice. And they collected weird mice, they collected naked mice, and anything they could lay their hands on. Well, Mr. Holman was a mouse fancier, and he had converted a garage into a beautiful mouse room, just as clean as you could believe. He built his own cages. They were beautifully made, and raised all these different kinds of mice. So we met him, and he was a really nice guy. And so he very kindly offered to give us some of his varieties, which happened to be some of the ones we wanted. By no means all of them, but a large percentage. And he didn't like the lab, because the Jackson lab, because they were kind of offhand. They got some mice from him, and they never really properly thanked him. And so we were very appreciative of, particularly Bill was an amateur woodworker, so he appreciated the boxes Mr. Holman had made, and Holman got very kindly disposed, and we got a lot of mice from him. And then after that, different well-known research facilities. Eventually we got some standard strains from them too, but it took a little while. And we had at that time at X10, which was the main location for the then called Clinton Laboratory. They gave us a small wing of the cafeteria building, and one room in that small wing became our mouse room, with just wooden shelves, and that's where we started constructing the stains that we needed. Yes. But we moved out of there into the Y12 facility, which was the electromagnetic method for separating uranium isotopes. And Dr. Hollander, Alex Hollander, was a great go-getter, and he managed to latch on to three, no, at the time it was two, it became three adjacent empty buildings, and we moved into one of those, and that became the mouse house, and then the rest of the division moved into the adjacent, much larger building. And to start with, they gave us the ground floor of that 9210, which became the mouse house. And it was going to be used during the war for a special separation method that never came to pass. So it had big machinery. It was completely taken up with huge machinery that was so heavy that the floor had to be raised in little concrete pedestals. So a long period was spent just getting that machinery up, and the pedestals couldn't be removed, so the rest of the floor was raised to be at the height of the pedestals. So finally that first floor was empty, and it had a really special air conditioning. It essentially used HVAC filtration because it was very important to avoid any air contamination, pollution of any kind, which then could change the spontaneous mutation, so-called spontaneous mutation rate, and we wouldn't have a very good baseline to compare the induced mutations to. So for that reason, this huge air conditioning system was installed. And then all the mouse rooms were constructed. So altogether it took about a year and a half after we got here before we could move into this building in Y12. Yeah. And then we were on the ground floor for probably another 4 or 5 years. And then the second floor, they did the same thing. And then eventually, about 10 years after we moved in, we got the third floor also. And it became a very large facility that would house, I think it was 38,000 double cages. Our cages were all double. And probably a total of, they were never totally filled, but if they had been all filled, would have been like 200,000 mice. That was the capacity. Oh my goodness. Yeah. We had 66 mouse rooms, and each mouse room, I don't know how long it was, but it probably the size of a good living room in length, and had 4 aisles of cage racks in it, which were filled with the cages then. So Bill used a lot of his early time here was spent in just designing the cages, designing, the racks and the washing, everything. So that was mostly what he was doing. Designed a really, really good, well-working facility.
So you and Bill and your colleagues made a lot of important discoveries based on this research, and maybe you could talk just generally about what a couple of those were and why they're important.
Oh yes. Well, the main, I guess the main mission of the program, to start with at least, was to determine radiation-induced mutation rates. And before that, there had been very little work, done on anything except fruit flies and corn, maize. So those were the 2 organisms in which radiation-induced mutations had been studied. And trying to get something closer to man is why mice. And so that was the original prescribed mission, just to get radiation-induced mutation rates. Well, that was just the very beginning, but it stayed to be a big mission. And over the years, we essentially the main purpose became to study the factors that altered both the quantity and the quality of mutations. So over the years it became different kinds of radiation. To start with, it was X-rays and gamma rays, and then there's neutrons and internal emitters like plutonium and tritium, and the neutrons, for example, we also sent mice for above ground bomb tests, which at that time was still in progress. So, it was a range of radiations. It was also originally male mice of a particular type of germ cell development, not mature spermatozoa, but some of the cells that produce spermatozoa, because spermatozoa are out and gone. So whatever mutations are induced in them are of very temporary duration. So the idea was to study the cells that could accumulate radiation damage, that were just sitting there and essentially budding off cell lines that eventually produce spermatozoa. So that was the original cell stage. But then very soon we got to preparing different stages from those original cells, like about probably 10 different stages going to mature sperm, and then adding females at different stages. And I branched off into very early, because my mice weren't ready, I was branched off into a totally different area of research, which was teratogenesis, which is damage to developing embryos. So that wasn't genetic, it was a direct damage. It was developed because I was trying to do a genetic experiment on, not on germ cells, but on what we call somatic cells, just body cells. And in the process of working up the methodology for this, I got sidetracked into doing, because I found so many abnormalities, sidetracked into studying the effect on embryos. And so that, I was working on that probably about 5 or 6 years. It led to, I found that there were critical periods in the development. At certain stages of development, you would get certain very specific types of effect. Like, give you an example, like day 11 of an embryonic development, you would affect hands, arms. Day 11 and a half, you do legs and feet. So it was very specific critical period. So part of the work consisted merely in studying the time relations, getting the critical periods laid out. And then when it turns out that the first 4 days you got no abnormalities. You got only death or survival. The ones that survived were normal. But after that, starting from day 4 on, is when you started getting the major abnormalities. And so, we followed up the early period also. When we tried to equate the time range in mouse development to human development by picking, say, the same stage in humans where you get, I don't know, the heart developed in a human and the heart developed in a mouse, and plotting all those things. It turned out that it's the first 7 weeks in humans instead of, instead of 15 days in a month. That was the really critical for major aberrations, abnormalities. And as I say, the very early period gave no abnormalities, gave a lot of death, but the survivors were normal. And so anyhow, we developed this recommendations because at that time there was still a lot of medical radiation used, X-rays, just for diagnostic purposes. And so we recommended that if you could schedule, if it wasn't life necessary for survival, I think, if you could schedule radiations, it should be restricted to the first 2 weeks following a menstrual period. And that became known as some people call it a 10-day rule, and some call it a 14-day rule. Anyhow, it went into the medical literature, and it's still in use now. And so nowadays, every time a woman gives, because the problem is most women don't know they're pregnant. So just being asked, might you be pregnant, isn't going to help. And that's why we set a specific time range when it was safer to do the radiation, because ovulation occurs halfway between menstruations. So if you work between a menstruation for the next 2 weeks, there wouldn't be anything. And after that, for maybe a day or 2 or 3, would be the time when you didn't get abnormalities. So that became, pretty well known in medical practice, and it's still being used as far as I know. And so that was a Seidler, and a little byproduct was we also I worked out very quickly scored quantitative response that could give you could detect very small amounts of radiation to embryos, and so that became known as a quick test for somatic damage. Okay, so then I stopped the teratogenesis, more or less stopped it, although I got back a little bit, and went back into genetics research. And I would say probably the mid-50s. But then, besides the mutagenesis, there were so many, many other things that much more, basic findings that we went off on. For example, the mutations were essentially our tools, and the fact that we had so many mutations really was a help, because we determined pretty early that we would not only just count them, but we would maintain them, propagate them. So we ended up with hundreds of strains that propagated mutations, and they became great genetic tools for finding out a lot of other things. Eventually, combined with molecular genetics, gave us not only fine, small-scale genetic maps, but molecular maps, correlations of structure and function. And that was just Bill and me, but then we got many other people to join the program. And the earliest one, back in '51, I think, was Jean Oakberg, who who studied germ cell development. And that was very essential to know essentially what were our targets, what was the radiation what kind of cells was it hitting. And so there was, strangely enough, very little was known about the timing and the manner of particularly male germ cell development, which is very complex because you get all the successive stages. And that, after this doing the normal development, he also studied the effects of radiation on different stages of germ cell. So that was Jean Oakberg. And then we had, I guess, next had some visiting and temporary people, Bill Welchens, Ernie Chu, who was a cytogeneticist. In the 60s, we had, I may be wrong about the dates, but anyhow, we had Gary Shager, who was very interested in the metabolism of germ cells, and he also, things like the nature of DNA damage, and he developed some methods for scoring, what was he called? Anyhow, for different scoring of damage. And then later on, we had Bob Cumming, and in the late 80s, we fortunately got DOE's permission to hire some molecular geneticists. And then we really went to town getting all the genetic material studied by molecular methods. So that was just a very quick summary of the types of things that the mouse has studied. And
one thing I wanted to ask you about was about how you helped determine that the Y chromosome determines sex.
Oh, I'm glad you asked that. I'd almost forgotten about it. That actually did not require the radiation. We found a spontaneous mutation that we called scurfy. And it was a weird, it made a mouse look crummy, sick. And to start with, Bill was just throwing these out because he thought, this is a sick mouse. Pretty soon appeared that they were all of them were males. So it turned out to be actually the first sex-linked mutation in a mouse. And because these crummy-looking males died before they could reproduce, Bill had to use a method he developed actually before coming here, which was ovarian transplantation from affected females, because once in a rare while, we'd get not just males, but occasionally a female. And after quite a bit of study, it turned out that these females carried only one X chromosome with a mutation on it, because there was an equal number that carried one chromosome without the mutation. But so you had females that had one X and nothing else. The males that had the one X and a Y. And so it turned out that XO, which was one X and nothing else, was actually a female, which was different from Drosophila, where XO was a male. And with the evidence from those XO females, it was easy to conclude that the Y chromosome was male determining in the mouse, which was also different from Drosophila, where male is determined by the balance between the number of X's and Y's. So one single Y, I mean, if there was a Y present in a mouse, it's always a male. But if Y in a Drosophila, it's not. It depends on the ratio of that one Y with a number of X's. And so that led us, the finding of the what's called XO females led to, along with the finding of other abnormal sex chromosome combinations, led to the discovery, to the conclusion that the Y was male determining. And the other thing that was very interesting, just about that time, a little later, we found what turned out to be rearrangements between an X and what we call autosome, which is a non-sex chromosome. So they're sticking part of an X to part of an autosome, and it turned out that the genes on that piece of autosome got inactive whenever the X was inactive. And it was just first near the junction, but spreading to a certain distance from the junction. So these were called the X autosome translocations, because normal translocation, ordinary one, are between two autosomes, piece of one joining piece of another, and reciprocal, the other piece joining that piece. But with the X autosome translocations were discovered because the genes on the autosome were inactivated, and and this was found in a mottled mouse. It wasn't the same color all over, just part of it, and that led to the conclusion that only one X is active. Even though every mammal has two X's, only one of the two is active. So, and which one it is random. So every female is actually a mosaic of two different kinds of cells. Some cells in which this X is active, and some cells in which the other one's active. So that again led to a study of what we call mosaicism, that is, animals that have, different genes active in different parts of the body. And it was a very fertile field.
One other detail in the article you mentioned was something called it was a chemical called ENU.
Yeah, we used a lot of that. That didn't happen till probably the 70s. We got into testing chemicals. Some of that was wished on us. Essentially we were asked to do. We used the same old methods that had been used for the radiation mutagenesis, and I don't know how many chemicals, probably about 30 or 40 altogether, maybe more, that we studied. Not all of them to the same extent, but one very interesting thing that came out of that is that one chemical called ethyl nitrosourea turned out to be a super mutagen. It induced huge numbers of mutations. Not only that, but they were all single locus mutations. Instead of breaks, or instead of cutting out a piece of chromosome like radiation does, they made just one spot mutation. And so that became very soon used pretty worldwide by people who just wanted to make mutations. This was a great method for making them. Normally, they're pretty rare. So ENU is the abbreviation. So ENU became a mutagen of choice for making mutations. Yeah. The other thing I meant to tell you, nothing to do with chemical mutagenesis, but well, it does in a way. I told you when I first got here, I was trying to develop a method for measuring mutations in somatic cells, and I got sidetracked into the embryo work. Well, after I got sidetracked, I went back to the original experiment, and that eventually led, well, we used some of the same markers, which were external markers, color, ear size, used some of those same markers for the somatic mutation. And because there's so many more targets, you can pick up a mutation or in many more cells. It turns out like day 11 was the, I think it was 11, was the optimum time for inducing a mutation that was easily visible. See, if you have too many cells, you have an embryo, you make the mutation in one cell. As the embryo grows, the mutation, the cell multiplies, makes a spot. Well, if you do it when there are too many target cells, it's not going to grow that much more. It's going to be a very small spot, might not be visible. If you do it too early, there'd be so few target cells that you wouldn't have that great a chance to find it. So the optimum stage for that, if you do it at the optimum stage, you get a spot maybe this size that you can easily see. And that got developed into what we call the spot test. And it became a very favorite, it's a short-term test. It's a genetic test because it measures mutations, but not in a reproductive cell, but in a body cell. So the spot test was another method we had developed here.
One other question I had was if you could talk a little bit about some of the technicians and caretakers that you worked with.
Yeah, first of all, we had animal caretakers. When we were still in that single mouse room at X10, we had one animal caretaker. Eventually, this built up into, I think, 17 or something close to 20 caretakers. And they would do things like once a week, all the mice got transferred into a clean cage with clean shavings. Then also once a week, but on a different day, the bottles got changed and washed and sterilized and stuff. So that's the kind, and then they had big machines for washing the cages. They had bottle carts. So that's what the animal caretakers did. And as I say, that built up to almost 20 when we had a very large population of mice. But on top of that, we had technicians, and the technicians were doing, the treatments with radiations or chemicals. They would score the mutations. They would breed different mutant stocks, and they were almost all bachelor-level college graduates, and a few of them were masters. And to start with, they were all female, which was amazing. And they were all, at that time, at the early time, many of them came from the southern women's colleges. And you wouldn't think that was a likely job for these girls to take, work in with dirty mice, but they were great. We had one woman by the name of Gloria. She was like somebody out of Gone with the Wind, exactly like that. Well, she developed this great method for catching mice. If a mouse gets on the floor, if you drop it, it gets on the floor, it goes all over the place, just that quick, And they're hard to catch, particularly because there's a rack that goes down the center of the mouse room, they would get to the other side by the time you were over there. He'd go back. So Gloria developed this wonderful method. She would spread out her lab coat and crouch down, so she made a tent over the mouse. And the mouse got very quiet because it was dark in there and wasn't scared, didn't run around. She could just grab in there and pick him up. So that was, and it was particularly wonderful because, as I said, she looked like somebody out of Gone with the Wind. We had a great group of technicians, and they were very compatible. They had, you know, it was like a social club almost. They had parties and get-togethers and things like that. And then, of course, we had some of the senior investigators. Eventually, I think, we had 2 males, but that was pretty late. But for a long time, they were all women. So that was a good group.
At this point, was it called the Mammalian Genetics Division? Is that right?
No, it was a section in the biology. It was a biology division, and to start with, there were, I think, 3 sections, and mammalian genetics was one of them. And things changed over the years. After Dr. Holland retired, there were different division directors, and one of them had the great idea, no, I was wrong, there were 6 sections, had the great idea of combining some of them. And so all of a sudden, I was the section head. I ended up with, instead of like 8 PhDs, you know, and lots of technicians, I ended up with over 100 PhDs because some of these other sections didn't have many technicians, but had mostly senior researchers. So I had this huge group of senior researchers. It was very unwieldy. All of a sudden, instead of a lovely, cohesive, friendly group, there were all these jealousies and problems. And eventually, maybe 3 or 4 years after that, the next director was wise enough to split us up again. So we went back, became mammalian genetics. It wasn't always, there was always something attached to it. It'd be a mammalian genetics and reproduction, or mammalian genetics and development, but always mammalian genetics to start with. Yeah.
So shifting gears a little bit, what do you think Oak Ridge's legacy is for human health and understanding the effects of radiation?
Well, they did the whole area of radiation risks, genetic risks, and you know the risk, the doses, permissible doses, were set by committees. There was a UN committee, an international committee called UNSCEAR, UN Scientific Committee on Atomic Radiations. Then there was a National Academy American committee. So between those 2 committees, radiation permissible doses for the whole world essentially were set by these committees. So they essentially used our data. That's what we supplied. You know, that the permissible doses were set on the basis of the data accumulated here, and had some interesting ups and downs because very early we found the mutation rate for those particular cells I told you about, the male germ cells that budded off eventually sperm, compared the mouse with Drosophila, which originally all the data came from. And the mouse had 10 times higher mutation rate. So all of a sudden, the permissible doses were lowered because it seemed like the risk was a lot higher. And then later on, we found that protracted irradiation, if you have a given dose, say 100 Röntgens, you can give it all in the course of a few minutes, or you can give it spread out over a year or so. The spread out doses were much less powerful than the acute doses. So that resulted, again, then they said, oh, we can raise the doses. So, it had its up and downs depending on, and then female mutation rates depended very much on the stage. So essentially the up-to-date findings had an influence on the permissible rates over time. I think they've stabilized eventually.
And you had mentioned that that Bill went to a nuclear test in Nevada.
That was wonderful. They eventually stopped above ground testing, but maybe it wasn't the last one. It was very near the last one. They tested them in the Nevada desert at a place called Camp Mercury. And so Bill and Gene Oakberg went in our old Ford and took a bunch of mice. And they were going to measure things that could be measured fairly quickly. One thing was what we call dominant lethals, which are mutations, mostly chromosome aberrations, that kill. And so that was one, kill the offspring, not the mice that have them. And the other thing, we were doing some embryological, irradiating embryos at a particular stage where it would be easy to measure the abnormalities. So they had to be, at the time of the bomb, they had to be at the right stage of pregnancy. So these mice were taken out in this old Ford, and it had in the back seat, there was a raised place for the axle or something. So they built a special wooden rack that fitted over that and was as wide as a car and had 2 or 3 layers, and they had mouse cages on that, and drove out to Nevada. And we had a wonderful cartoonist in our group who was the supervisor of the animal caretakers. He drew all these great cartoons of what would happen to these guys. Like they would have to carry these, every night they stayed in a motel. They waited until it was dark so they could carry these cages into their room, because it looked like a child's coffin, about the same size of them. Then they would carry it into their motel room and spend all night filling up the water bottles, because they couldn't keep the bottles on while they were driving, because they would leak. So he had cartoons about that and how they registered at the desk with the mice behind them. And anyway, they had this wonderful trip out to Camp Mercury, and the mice got exposed. The bomb went off on a tower that was built up above the desert floor, and then they had these, what they called hemispheres, which were lead, like igloos, 7 inches of lead thickness, to shield out all the gamma radiation. It was supposed to be just the neutrons from the bomb. And inside the hemisphere, they were like mushrooms stuck with a stalk down into the sand, and the hemisphere rested on the desert floor. And the inside was large enough to take a few mice, Drosophila, whatever. There were lots of other people there exposing things, and it was air conditioned in the inside. And they had these hemispheres set up at different distances from where the bomb tower was. And then they were out there like 2 or 3 weeks ahead of the actual test. they spent that time training how they could get this material out of there in minimum time, because everything was horribly radioactive after the bomb went off. So you could only spend like seconds or minutes was all you had to grab in there and get everything out. They trained to do that. And so when the bomb went off, they retrieved all the mice, and then Bill flew home with the mice in an old DC-3, I think it was an army plane, brought the mice home here the same day that the bomb had gone off. And, you know, they had to wear shoe scarves and special clothing. Well, when he got to the lab the next morning, he was told that he had to wear shoe scarves, and it turned out that the cloud with the fallout had moved from Nevada to Oak Ridge, and we had fallout rain, and people would measure radiation on their cars. They would put film, here in Oak Ridge, they put film on the car to see the spots of exposure that were made by radiation. There was a lot of fallout. So the mice, Bill and the mice were clean, but the cloud had come just as fast, almost as fast as he had. It's very interesting.
Wow, yeah.
But the mice were, you know, in good shape, and we could get all the endpoints. And then Gene Oakberg drove the car back. So,
wow.
The other adventure, totally unrelated, was that I had stayed at home because I had just had our second baby, and she was just weeks old. And I had my mother staying with me, and my mother decided she was deaf. The baby was deaf. So I would call Bill in Camp Mercury and say, "We have a deaf baby." Well, my mother turned out to be wrong. But, you know, all the time he was there. And one of the people that was also staying at the camp was a pediatrician. And so Bill asked him about that. And he said, "Who told you?" He said, "My mother-in-law." He said, "Pay no attention to mothers-in-law." But that was a sidelight of the bomb test.
One other question I had for you is the laboratory has the Liane B. Russell Fellowship now.
Yes, that was created about 3 or 4 years ago, and it is essentially for young women getting, you know, their own independent research facilities, because so many women who were just as educated as some of the men ended up working as a partner or technician, or, you know, not a high-level technician almost, to somebody instead of doing their own research. And so this is one way of getting young women their independent facilities and opportunities. And they're not all biologists, they're everything. They're physicists, mathematicians, and the first, they do 3 every year. Well, they have been doing about 3 a year. And the first 3 happened to be, every one of them was Chinese. It's very interesting.
Mm-hmm. What does that fellowship mean to you?
I was very honored. I mean, I'm not doing anything for it. It's my name. I meet with them, you know, when they first arrive. And once in a while I meet another one in between. But it's very gratifying.
I know you have a story as well about when you received the Fermi Award.
I received the Fermi Award in 95 or thereabouts, yeah. And some of our technicians went up to Washington to be there when I got it. It's very nice. And I received it I was supposed to see President Clinton, and he happened to be out of town on some function. So I got to see Vice President Gore. We went to his office, and he wanted my family being, well, my daughter was up there. She didn't know I was going. She was wearing some real old clothes, and they found her somewhere, and they grabbed her and said, "Come to Gore's office." So we have a picture. I have it right up there of Vice President Gore and Bill and me and my daughter wearing this crummy old thing, and yeah. But he was very great. He had been very, very helpful before he became, when he was originally a representative and then a senator. But even while he was still a representative, he came to the lab and he really stuck around and really wanted to learn. So many of the others came and it was just photo op for them. But he really wanted to learn. He stuck around and listened to people. So he was fairly familiar with our program. Yeah, we had some interesting visitors altogether. We had the Queen of Greece. We had President Carter. He was also very interested. But they got a program for him where 5 different people from all over the lab, physicists and earth scientists, everybody. And so I was the one that did the biology. And so 5 of them gave him little talks. And we got really rehearsed for that. But before that, he'd been around, and he also was interested. He was very, very bright, very scientifically educated, I think.
You've been involved in the environmental movement for a very long time.
Yes.
I wonder if you could say a little bit about how you got involved in that.
Yes. Actually, since 1965, what happened was, Bill and I had always, been outdoors a lot, and we had we'd done the Smokies, and we wanted to go in the Cumberlands. And a woman who worked in our building was married to a guy, a Dutchman, who was working at the lab, and he was a great paddler, and he had explored many of the Cumberland rivers, which are really whitewater rivers, solo. He'd just taken his own kayak and gone down and explored her. And he found one that he particularly loved, and he wouldn't tell anyone about it because he didn't want it to get developed. Well, she decided we were all right to be trusted. So they took the name was John and Barbara Bombay, and they took Bill and me on a 2-day canoe trip down the Obert River. They had kayaks and we had a canoe, and we camped overnight it's a beautiful river. It has huge sandstone boulders. It's in a sandstone gorge, very wild. So we camped on this huge boulder, caught our own fish, cooked them for dinner. And it was a great trip. And this was in June of '65. And almost Christmas Eve of that same year, there was a little item in the local paper, in the Knoxville paper, saying that TVA was about to build a dam on it. And so I hid the paper from Bill because I didn't want to spoil his Christmas. But then we looked at it afterwards and we were really upset. It was such a gorgeous place. So we got into trying to save that river. And we formed a group called Tennessee Citizens for Wilderness Planning in 1966. And it took till 1979 for the river to be protected. First we killed the dam idea, we did that, but we thought we had to go beyond that and get some positive protection. And so we tried to get it, well, first we passed, actually wrote and passed a Tennessee State Scenic Rivers Act, which passed. And the orbit was on it. There was about 9 rivers on it. It got amended off at the last minute. So then we tried to get it on the National Wild and Scenic, which was going through the Congress the same year. The orbit actually got on it in the study river category. You know, there were some instant rivers and some study rivers. So it was one of, I think, 30 or so, no, not quite that many, study rivers. And so after that, they had a long study, BOR study, that our group really contributed to. I mean, the group did a lot to kill the dam to start with, but then participated in the study in a lot of different ways. And BOR found the river to qualify for almost 100 miles. So that was wonderful, but then all sorts of local opposition arose. And we finally ended up, the story is too long to tell, but if you're interested, I can give you an account. Ended up with 45 miles, because being in a study category is not enough. You have to have a bill actually designating the river as a wild and scenic river. So we finally found a congresswoman who had only been in the job for about a couple of months because her husband was running for Congress, and he got killed in an airplane accident. And she took, she became a candidate instead of him. So she was totally inexperienced. She had to be really coached. But she eventually wrote the bill that passed in 1979. So this year, by the way, is an anniversary of the National Wild and Scenic Rivers Bill, which was passed in '68. So Nikki Nichols may have told you they're celebrating that this year at the orbit.
Yes.
Yeah.
So it sounds like it was quite a struggle for.
Oh, it was a major struggle, but that turned out to be just one of, because very soon after we had beaten the orbit dams, we got into a lot of other areas. The Big South Fork, there's another dam proposed for it, the Devil's Jump Dam. We killed that one, and then we worked for many years getting the Big South Fork National River and Recreation Area. That was a very complex struggle too. Ended up a lot bigger than it would have been if it had been a wild and scenic river. If it had been wild and scenic river, it would have been just a corridor, with a little bit up on top. But it ended up 125,000 acres with a lot of land up on top. And then we got into strip mine legislation and into other rivers and into the Appalachians in general. So TCWP's really tackling a lot of different issues. But the orbit was the first one, and that was since 1966 and it's still going.
Are there any other stories you'd like to share, or things that you think people should know about Oak Ridge and its legacies?
Well, Oak Ridge itself, one of the things we worked on was protecting the green belts, because Oak Ridge has a lot of green belt. And actually this happened even before we started TCWP. One day we saw this group of people just walking through here, and it turned out they were going to map the route of a major power line, 69 kV, it's a huge, and it was supposed to go on a turnpike. Well, they didn't want to mess up the turnpike, which is a big mess already, you know. It wouldn't matter to put it down there. They wanted to take it through the greenbelt. So, you know, we tried to find out what's going on. It was going to go up here and up to the top of the ridge and then run all the ridge for 9 miles or more, and then come back down to a power station down below. So this was actually our first fight. We fought that one, and before TCWP even started. And that ended up, again, not only did we squash the pollen, but we also wanted some positive protection for the greenbelt. So we thought if we developed a trail up there, you know, people would get to, most people didn't even know it was there. You would get a constituency in favor of keeping the greenbelts. So we developed the North Ridge Trail, which is 11 miles long and goes all the way from the east end almost to the west end of Oak Ridge in the northern greenbelts. And it's just a walking trail, just wide enough for 1 or 2 people, you know. It's a lovely trail, you know, it goes up and down into little creek beds, and it's already fulfilled its purpose, because there have been other threats. Somebody else wanted to build a road through to a development, so we managed to beat that. And altogether, greenbelt protection became an Oak Ridge issue, which it has been since 1965. So that's one way we got involved in Oak Ridge.
So what issues are you working on today with TCWP?
Today?
Yeah.
Well, I'm among other things, I do the newsletter, which comes out every 2 months, and it's quite extensive. I'm going to give you a copy, and so that takes up a lot of my time. But other than that, you know, I just participate as a board member. Not nearly as much as I used to do. I used to do a ton, and Bill did too. I mean, Bill was essentially, you know, he was forcibly retired in 75, at the age of 65, because at the time that was retirement age. So after he retired, he became a half-time consultant, and so he had a little time to do things, and he was immensely involved in trying to beat the Teratogenic Dam on a little T, because that was a lovely river too. Teratogenic Dam, I mean the little T, not successful. And the other thing he really fought hard was the Duck River. There are 2 dams, Normandy and Columbia, and he was fighting, at that time they were doing the Normandy. That was the worst, the most unjustified dam that was ever built. But he lost that one too. But that was one of the things that we were doing too. So that was really sort of a heartbreaker. And another thing we're involved in is trying to get more wilderness designated in the Cherokee National Forest. That's really weird. Both senators, Senator Alexander and Senator Corker, have been sponsoring a bill ever since like 5 years ago, the same bill. Can't get the House to support it. So that's a current issue. And we've been concerned with things like coal ash basins at the TVA power plants, and you know, and that took a step forward and it's now taking another step backwards, because finally they passed a good regulation. Of course, the current EPA just threw it out the window. And you know, that's a real problem, because the coal ash has a lot of toxic agents in it. and they just store it in ponds, which are most of them not even lined. So you get groundwater leaching out of those ponds. And was it in 2008, at Kingston, they had one of the ponds burst, and they got all this coal ash to spill out all over the land there. And it's a major problem because it's all over the country, the coal ash ponds. So that's another issue we're in.
One other question I just thought of, and this is totally going back very far, but I remember reading that when you were when you had to leave Austria, you had to hide or get rid of your books. Is that right?
As soon as the Anschluss happened, they were searching for what they considered subversive books. So many people who had a fireplace would burn up the books. But we didn't have a fireplace. So we flushed them down the toilet. We'd tear out the pages because we couldn't put too much down or you'd plug the toilet. And so my sister and I were spending days just flushing books down the toilet. That was interesting. Yeah. But it turned out that the reason we were able to get out so early was because one day my parents were out, just my sister and I were there. This knock on the door, this gigantic SS guy, he was like 6'4" something, in his black uniform, knocking on the door, looking for my dad. And eventually, he wanted to take over my dad's business, which he could have done any of, but my dad was representing some chemical plants from England, companies from England, and maybe some other countries. And he wanted to take over those representation things, whatever they're called. So he was trying to play nice. And he wanted to send my dad to England to talk to them. So he said he wouldn't go unless we all went. So we all went. We spent probably 3 weeks, packing. We thought everything would get moved over there. So we'd pack things carefully, wrap things like glasses, everything. Never came. They never sent it, which is all right. So, but that was it. It's because of him we were able to get out that quickly. So it turned out for the best.
It's fortunate in a way. Yeah.
It's very interesting. My sister, way later in the 50s, she went for a visit to Vienna, and she went to see this guy who had taken over my dad's office. And in his office, he had, from our apartment, he had got rugs from the apartment, he got pictures hanging up on the wall. It's interesting.
One other interesting story I'd read about is that you got to go to East Germany in 1981.
To Germany? Oh yeah, that was fun. The spot test I told you about. They had a lab in East Germany, I've forgotten the name of the place, that wanted also to do the spot test. So they organized a meeting devoted to spot tests. So I was visiting somebody who had worked here, actually thought he was going to stay here, but eventually went back, Udo Ehling, and he had set up a mouse genetics lab near Munich. And I was visiting him, and one of his collaborators, who is still a good friend of mine, was going to drive to this meeting in East Germany. So she was going to drive me. And we drove up there, and she took along a trunk full of coffee, sugar, because they didn't have it. And the host up there was going to have a little party. Couldn't have a party unless she brought all these foods to him. So we drove up to, it was just a long day's trip, but getting across the border was fascinating because there was all this no man's land between the two fences, like from here maybe to the other side of the valley. And we would hand them documents on the West Germany side, and they would shoot them through a tube to the East Germany side. And after we got all cleared, we were able to go across there. But coming back out of East Germany, they searched our cars because at that time there were a lot of people trying to leave by hiding in other people's cars. So they would open the hood. They had these long spikes with sharp points. They poke it all between the parts of the engine to make sure there was no one underneath. They wheeled mirrors under the car. Boy, it was amazing. And East Germany was very depressing. It was very austere, the store shelves were empty, the road surfaces were horrible, and it was just, the lights were hardly on. It was just a very depressing place to be. Interesting visit. When I got back, I was going to be, what do you call it? I was visited by a CIA guy who wanted me to tell him all I knew about East Germany, because I didn't know very much, But he had all these questions about science, which all I knew was this particular meeting.